to our STORK seminar, first of the spring series, which is both exciting and depressing that it's already spring. But today we've got Richard Lopez from Bard College, who is going to talk about specification curve analysis and how it can potentially be used to promote more transparent and robust science. So a topic that I'm definitely very interested to learn more about, but as I've just admitted to Richard, I know almost nothing about, so really looking forward to this. So thank you very much, Richard, for coming today, and whenever you're ready.
Thank you, Emer, and thank you so much to this group. It's a pleasure to have this opportunity, even virtually, to share a little bit about a new method to me as well. But just to give you a quick introduction about myself, just so you know a little bit more about me. My background is in psychology and cognitive neuroscience. And for my postdoc, I received training in translational neuroscience and thinking about the impacts of our behaviors on various aspects of mental and physical health. Here at Bard College, I teach a variety of courses, including intro psych, social neuroscience, a seminar on the science of goal pursuit, as well as a hands-on lab course. And even though it's a small lab, I have a fearless group of undergraduates who are helping me conduct research in my regulation of everyday affect, craving, and health lab, or the REACH lab for short. And it's really a pleasure to connect with researchers in other communities, because I'm clearly not trained in kinesiology or sports sciences like many of you are. So if anything today sparks an idea or potential for collaboration, I would more than welcome that. So feel free to email me or connect with me on Twitter or LinkedIn. So just to tell you a little bit about my research program, to date, I've been very much interested in identifying neural indicators of various self-regulatory behaviors, especially in the eating domain. I've also looked a little bit on media multitasking as kind of behavioral phenotype. Or risk factor for self-regulation failure, and how people who are high media multitaskers may process external cues a little bit differently, and looking at individual differences along that dimension. Although, kind of encompassing all of my research and kind of thinking about how I will take my research going forward, I'm very keen to use various meta-science tools to improve the quality of my own science, but also to kind of keep abreast of a really kind of changing landscape in terms of methodology that's available to us as scientists. And today I'm going to talk specifically about specification curve analysis. And there is one paper in which my collaborator and I used this type of analysis, and I'll be getting to that later in the talk. So for today's talk, I want to kind of give a brief overview and history of multiverse approaches, and then do a quick primer on how to conduct specification curve analysis from a conceptual level. I'm not going to get into the nitty-gritty of carrying out the steps in R, but I will point you to some resources to do that, but just kind of getting us on the same page conceptually on what SCA entails. And then I'll close with a case study from translational neuroscience, so more close to my own research on how my collaborator and I used SCA to identify robust brain behavior associations in the eating domain. So let's start with a quick history. One concept or phenomenon you may all be aware of is researcher degrees of freedom, which refers to undisclosed or opaque analytic and or interpretive flexibility when conducting research. And this kind of the proverbial what hit the fan when this paper came out in 2011 by Joseph Simmons and others, titled False Positive Psychology, Undisclosed Flexibility in Data Collection Analysis Allows Presenting Anything as Significant. It really shook the psychology world in a lot of ways. But why are research degrees of freedom problematic? Well, it's not quite clear which analysis decisions were made by the researchers a priori versus post hoc. And this could be quite a challenge if you're trying to either conduct a conceptual or direct replication, right? You just don't have that information available to you. And perhaps more troublingly, researcher degrees of freedom can result in higher probability of false positives. So I just want to bring your attention to this one column in this table from the Simmons et al. Paper. And going down row-wise are kind of different scenarios where different things are toggled in terms of controlling for different factors. And you can see the false positive rate is anywhere between around 10% to as high as 60%. So this is really, this is quite problematic. And more generally speaking, with regards to researcher degrees of freedom, you have this, this idea of the garden of forking paths. And this originally comes from a novel by Jorge Luis Borges, El Jardín de Senderos que se bifurcan. And what you have here is so many steps in an analysis that a researcher does. There are so many steps where you could tweak the data or look at it this way or look at it that way, look at it this way, look at it that way, in terms of binary decisions. But there are so many points, decision points, that we have to kind of confront, right? And some of you may be familiar with this Robert Frost quote, "Two roads diverged in a wood, and I took the one less traveled by, and that has made all the difference." But with potentially sketchy research practices, you could have people either knowingly or unknowingly, wittingly or unwittingly, making decisions that result in statistically significant results so their papers could get published and so they can secure more grant funding, et cetera. Now, in my own field, in social neuroscience, social and cognitive neuroscience, the garden of forking paths is even more fraught. So generally speaking, if you have X binary decisions, you have 2 to the power of X possible paths, but often it's not just 2 options, right? So for every decision point, you may have more than 2. And Russ Poldrack and others kind of sketched this out in terms of different analytic pipelines when analyzing fMRI data, which are multidimensional. There are a lot of ways to clean the data and filter the data, et cetera. So they have this very extensive table in their paper, kind of outlining every processing step and the number of plausible options that you could do within that processing step. So in this case, for motion correction, you might have 4 plausible options, for slice timing correction, 3, and so on. And remarkably, if you kind of do all the combinations of possible ways to analyze the data, the total possible workflows is as high as 70,000, which is just really incredible. So this is an issue. But what about pre-registration, right? So pre-registration has become in vogue, which is great, and it could help constrain the space in which we make our analytic decisions. But there's often value sometimes, right, in trying out different model specifications. What if we include this set of covariates in the model versus that set of covariates, right? And you're trying to pre-register all this ahead of time, but it could be quite unwieldy. And there are just multiple reasonable ways and paths to analyze the same data set. But I would argue that if you have too narrow of a pre-registration, it can inadvertently result in false negatives because the model specifications cover too small a patch of modeling space. And going back to what seems like real, ages ago, Donald Rumsfeld famously said there are unknown unknowns, right? In the sense that, if we're conducting an analysis and we're carrying out a very, very specific model specification, we may be missing out on really interesting associations or effects. And we might be shining the light in terms of our analysis in just too small a patch of modeling space. So drum roll, please. Enter the multiverse. Does anyone know where this screenshot comes from? From which movie? Does anyone want to shout that out? If you've seen this movie. Interstellar, yes. So this is the part of the movie. It's very epic, dramatic, and very heart. A lot of heart behind it too, given his relationship with his daughter, where he enters the Tesseract, this multi-dimensional space. So, what is the multiverse? What comprises the multiverse in terms of how we collect and analyze our data? Well, Stedian and others laid out the fact and just kind of highlighted the fact that there are many ways that our raw data in our studies can be cleaned, computed, and operationalized, which can lead to a data multiverse. But importantly, there are many intermediate data processing steps that can lead to a multiverse of statistical results, including p-values. So depending on the effect of interest and really if it's a real effect or how strong of an effect it is, if you plot the p-values, in this case, like Stegen and colleagues did, you can get very infrequent significant effects of interest, or you can have the effects kind of skewed in such a way that most of them, or at least more of them, are significant. And Stegan and colleagues say also that multiverse analysis, its utility is that it helps avoid, or at least reduces, the problem of selective reporting by making the fragility or the robustness of results transparent. And this can help identify the most consequential analysis choices, right? So, in 2016, these issues were being brought to light, and Uri Simonson and others had a paper that just came out last year to kind of lay this out a little bit more systematically. And Simonson and colleagues say that it's really about the model specifications. It's a really helpful way to think about the multiverse of data and the analyses we do. And model specifications necessarily adhere to a hierarchical or nested structure. So the superset is a very, very large, potentially infinite number of model specifications. But within that superset, you have really important subsets that have to be defined. The first subset is a set of valid specifications, right, that would be feasible to consider. But then within that, you have a set of reasonable kind of non-redundant specifications. And that hinges on how a particular researcher conceives and makes assumptions about the modeling space. So Simonson and others lay this out in a figure, just kind of schematically, where you have all possible specifications in the largest oval, and then you have valid specifications in the blue oval, and then you have non-redundant specifications in the innermost red oval, and then you're selectively reporting, maybe just a few models at most in just that little patch of modeling space, right? But again, this hinges on how a researcher thinks about the modeling space and which measures and variables should be tested within that modeling space. And if you have another researcher who has similar views and you ask them to kind of map out all the possible models and the subsets in the space, you might come up with a pretty similar kind of scenario here. But again, they may report other models within kind of this non-redundant set. But what happens if you have researchers with different views? Well, they could not necessarily agree on which parts of modeling space that they're looking at in terms of how they set up their models, which variables they look at, et cetera. So this is kind of all the kind of background knowledge and rationale behind specification curve analyses. And now I want to kind of go through a very quick and by no means totally comprehensive primer on how to carry out SCA, how to carry this out. And I should say, I'm relatively new to this, probably the last year or so, and I've used, being inside a lot with the pandemic and everything, just as opportunity to kind of marinate in this method for a little bit. But I'm still quite new, but I'm kind of sharing what I know thus far, and it's a method that I hope I continue to use in the future. So there are 3 steps. All you need is 3 steps to conduct specification curve analysis. The first one, which maybe in some ways could be the least trivial one, is to determine a reasonable and justified set of model specifications with all of your analytic decisions and options clearly stated. So ideally, you would do this ahead of time, before you collect, or if you have the data, before you analyze the data at least. Then you would generate a specification curve showing an effect or effects of interest across all the model specifications. So again, that's kind of one of those smaller ovals in that figure I showed you a little bit earlier. And then you can run not traditional, but a slightly modified kind of inferential statistics to establish how robust, how reliable or consistent is an effect or effects of interest across all the specifications, right? So these are the overarching steps that will, that you have to kind of carry out if you're going to do any type of specification curve analysis, generally speaking. And these are steps that Simonson and others kind of lay out. But I want to kind of walk us through an example, and this is an example in psychology research, and it's and I have to give credit where credit's due. My collaborator and friend, Dr. Danielle Cosme, she set up a wonderful tutorial on this, on how to kind of work through this example with SCA in R. But I'm just going to kind of hit on the highlights here according to the 3 steps I just shared with you. So the first step is to determine, like I said earlier, a reasonable and justified set of specifications, where you kind of lay out all the different options and decisions that are toggleable. And in this tutorial example, the research question is, what is the relationship between mental health and life satisfaction? So that's kind of the topmost research question that will be addressed with this specification curve analysis. And as far as the most important analytic decisions that will, of course, affect the number of specifications is, well, what is going to be the primary predictor of interest here? So there are actually 4 possible ones. There are 4 candidate predictors. There's depression score, anxiety score, a global assessment of negative affect, and perceived stress. And then there are several control variable here, or covariates, that could be included. And in this data set in particular, in this tutorial data set, there are 3 possible ones: age, gender, and maternal education. There could be others. But I should say, just while we're on this point, that I think it's really, really important with regards to covariates. This is just like a separate point, a side note, that you should have good reason. You should have reasons for every covariate in your model. I caution against just throwing random sets of covariates in and out. There should be reason to either control for those variables with regards to what's been done in prior literature and prior work. But then also, if you have reason to believe that these are already established predictors of the outcome measure of interest, and you want to control for them because that's already known, and you're identifying maybe a new predictor of the outcome. So I think it's really tricky to decide on which set of covariates you include. But at least in this case, there are only 3, just for the purpose of the tutorial. And then there's yet another analytic decision here, and that is, how do you handle outliers? And in this case, there are 2 possible ways to handle them: either use all the data points or Winsorize the data within plus or minus 3 standard deviation, right? So if we kind of visualize all the possible model specifications with just a few of these analytic decisions, you can see it kind of really expands, right? Because you could either have, you either Winsorize the data or not. Which of the predictor variables are you going to use as your primary predictor variable? And then in each of those models, which of those, which sets of control variables or covariates will you use? So this is just kind of the visualization of this, just laying out the total possible numbers. So what does a specification curve look like? Okay, so we have to generate the curve, and there are different ways to display results from an SCA, but I'll start with kind of the most common, right? And just before I show you the figure here, it's important, and it's taken me a little bit of time to learn how to read an SCA curve. So if you're going down row-wise, or with, if you're going down, just down generally speaking, you're looking at a particular effect of interest given certain analytic decisions you made. So you're actually looking within a model specification. So every line that you go down in the specification curve, that's a specific model specification where you're looking at an effect or association for a particular variable of interest. And it's kind of giving you a snapshot. Oh, what if I decided to do this, 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 and this, right? It's a snapshot of one scenario, one specification. That's going kind of down in the curve or graph. But then if you go left to right, that's when you can start getting a picture, at least qualitatively, of how robust and consistent is the effect of interest across model specifications. And in some cases, there may be hundreds of model specifications, maybe thousands. In this case, because it's a toy tutorial example, it's not as many as that. So you can kind of see everything clearly. But again, going left to right, that lets you kind of look at the consistency of the effect overall across model specifications. And going up and down, you're kind of traveling within, a particular specification. So this is what the curve looks like. So you have your specification curve just indexed here from 0 to, I think there are 64 or so total possible specifications. So that's what's going, what you see on the x-axis. And then usually plot some sort of effect size or standardized measure of effect size, in this case, the standardized regression coefficient, because this is regression. And So you usually have that top panel, but then coupled with it, you have the bottom panel, which kind of lays everything out really, really clearly. Now, it's a lot to take in, but like I said, if we just remember how to read this up and down, left to right, it's a little bit more palatable. So if you go up and down, right, you can kind of go into a particular specification and say, okay, which predictor was used? Was the data Windsorized or not? What were the covariates, right? So you kind of see kind of this a particular set of decisions that are represented by that model specification. And I should say the red and the black colors here indicate whether the primary effect of interest is significant at p less than 0.05. So anything red here means that it was significant at that threshold. So what you can see is that as you go kind of across, right, you have these effects of interest. Well, this is the overall effect here, but critically, you could see that if we line up these effects of interest here, we could actually see depression is most strongly negatively correlated with life satisfaction, which was the outcome measure, followed by perceived stress, followed by anxiety, like they're all negatively associated, right? But then the kind of global negative affect measure, it's most weakly associated. In fact, in all of these model specifications, the confidence intervals cross zero, and overall, the effect is essentially null. So here we could see that perceived stress, anxiety, and depression are significant predictors of life satisfaction. Significant negative predictors have a negative impact on life satisfaction. And this is across the modeling space, where you can kind of make that inference. But this was kind of qualitative, right? I mean, you can talk about the graph, you can talk about kind of the overall patterns, but how do we quantify this a little bit better? Well, that's where step 3 comes along. And here, there are different kinds of inferential statistics you can use, but again, not so much the traditional inferential stats. It's a slightly modified version with bootstrapping. So the idea is you run your SCA, you extract the median effect size, and the number of models with the statistically significant effect of interest, right? So that's kind of what I've showed you already. But importantly, right, because we're not doing traditional NHST, we're not comparing the inferential stat with kind of the idealized hypothetical distribution of sample means. Here, we actually need to create a distribution of SCA curves under the null hypothesis. So you actually go in and you remove the association, any association between X and Y to kind of generate your null distribution. So that's your kind of reference distribution, because you're trying to see how extreme or significant is the effect in the observed SCA versus the bootstrapped SCAs that don't have any effect in them, right? So that's the key step. And then the last step here is you count the number of samples in which the curve metrics, such as the median, were equally or more extreme than the observed metrics to generate p-values indicating how surprised the observed results are under the null hypothesis. So here, just kind of looking at all the IVs, overall they have a median negative effect, negative association of negative 0.31, and a more extreme median effect across all the model specifications in the bootstrapped samples, where you're just resampling the data, where there's no effect. It happens very rarely. So that's kind of the two-tailed version. And then all the other effects are either are essentially positive, and it's again very rare that you'd see a more extreme effect. So these are kind of the one-tailed stats that come out of this. But if you just look here in these two columns, you can say, okay, the median standardized regression coefficient for these independent variables of interest is negative 0.31, and it happens so, it's so rare, very, very rare, that you see that effect in the null distribution. So you can conclude or infer that these independent variables are indeed reliable predictors of life satisfaction. So this is kind of just a toy example, like a tutorial example, like I was saying, but all of these steps, and just going back a second, all of these steps would be the same regardless of your particular analysis scenario in terms of the data you'd be working with. So again, you determine a reasonable set of specifications. You lay out what your different analytic decisions or options are. You generate your specification curve, right? So that gives you a really good overall qualitative picture. And then you just do some additional bootstrapping, create a null distribution and compare the observed SCA curve against the null SCA curves, just to see truly how kind of significant and strong the effect is in the observed SCA. So that's the overall idea. And I should say there are actually some really fascinating additional plotting schemes and options that you can utilize when you have data from a specification curve analysis. One way to do this is to kind of plot the ranked effect sizes of your predictor or predictors of interest across the specifications. And you can kind of compare these effect size traces as a function of other analytic decisions. And Orban and Przybylski in 2019 did this when they were looking at relationships between adolescents' social media use and phone use with different measures of well-being. So you can see here, they've ranked the specifications based on the standardized regression coefficient, and they have kind of two traces. They have one trace where they didn't control for any other variables, and they have another trace where they did control for variables. So you can kind of visualize the effect across modeling space. And this is important if you really want to highlight one particular effect of interest, or maybe multiple effects of interest. But again, you're able to kind of add these other factors into the plot to say, okay, we're interested in a particular effect, but we want to see how these other analytic decisions might impact that. So that's one way to do it. Another plotting scheme that's becoming more popular is you plot kind of the surprise value, which mathematically is the inverse, the negative of log base 10 of your p-value. So you take your p-value and then you take the negative log base 10 of your p-value. And in this case, higher numbers are better. So it kind of indexes surprise. So you plot surprise value, which is calculated from that, higher is better, against the effect size, for example, betas. And this is what's known, I really like this, it's just known as a vibration of effect plot, a VOE plot. Interestingly enough, this is actually quite similar to volcano plots in genetics research, where you're looking at many, many thousands potentially candidate genes and their effects on other variables of interest. And Del Giudice and Gangsted, this paper just came out that talks about multiverse analysis and various considerations with it. They plot here, and this is from simulated data to kind of make a point they were making in the article. But here on the x-axis, you have effect size, and here on the y-axis, you have, again, that surprise value, where higher is better. So here, with the dotted red line, that's just giving you a reference point of p of 0.05, right? So here in this case, like, again, this was simulated data, but you could see that the effect here, there's no effect. It's all over the place. You have a lot of values that are close to 0 and some extreme values, and a lot of the model specifications include effects that don't really meet that threshold of p of 0.05. But this is just another way to kind of quantify results from a specification curve analysis, just setting it up a little bit different. But I wanted to commend this to you. You may see this in papers that use it. Okay, so in kind of the last part of today's talk, I want to talk about one study that my collaborator, Dr. Cosme, and I did, where we wanted to identify robust brain behavior associations in the eating domain. But before I go into that, I want to say, and maybe some of you have noticed this, but researchers are indeed beginning to adopt and leverage SCA to help improve their science and to get a better sense of the effects that they care about. So Julia Rohrer and others looked at this a few years back. Like I said, Amy Orban and Andrew Przybylski have looked at this in terms of adolescent digital technology use and associations with well-being. And now, at least in my area of research, kind of cognitive translational neuroscience, I've looked at this in terms of different neural indicators of eating behaviors. So I kind of want to just lay out how my colleague and I leveraged specification curve analysis to help us address the questions we had in our study. So broadly speaking, there are different ways you can research self-regulation of eating, but in a lot of the studies I've done in the past, I've combined neuroimaging with ecological momentary assessment, because that allows for kind of establishing associations between patterns of brain activity and people's momentary and daily eating behaviors. So for those who are not familiar, there's a kind of task known as a food cue reactivity task, where you bring people into the fMRI scanner, you show them various images, and you give them an incidental task to complete. In this case, the cover story was we're interested in kind of perceptual and spatial processing, so just categorize each image as taking place indoors or outdoors. But we sneakily inserted images of appetizing high caloric foods into the stimulus set, so that at the end of the day, we would only look at their brain activity in response to those appetizing food cues compared to all the other images. So that would kind of generate a brain-based kind of signal or index of how their brain is responding to the food images. We also assess participants' body mass index and aspects of their body composition using biological impedance. And last but not least, and critically, we also have participants complete an EMA protocol where their different aspects of their daily eating behaviors are assessed, including how frequently they gave into desires to eat food in their daily lives. And that's usually takes place for about a week, sometimes longer. But the key piece here is how can we relate different neural indicators of their responses to the food cues to kind of the proximal aspects of their eating behavior as assessed via EMA, and kind of what we're thinking about as a kind of a distal, kind of broader index of their eating as indexed by BMI and adiposity in terms of percent body fat. So to help anchor us kind of theoretically, we used a newer model in thinking about how people regulate their eating behaviors. And I'm just calling it a tri-process or three-process framework. Now, traditionally, if we're thinking about, okay, we have an unhealthy food, then we might have reactivity to that food, which will impact our choice. And if we're experiencing kind of more motivation and desire for that food, we're more likely to eat it. And then maybe regulation kicks in and kind of dampens the reactivity or dampens our urges, and then we're less likely to eat the food. So that was the framework for a long time. But in this framework, you actually have three critical processes at play. You have reactivity, you have regulation, and then you also have this orthogonal or relatively orthogonal construct of value, how people are valuing, do they value regulation or do they value eating healthy or unhealthy? And that's going to affect their eating decisions as well. And it turns out, in terms of the neural correlates of these three processes, reactivity, regulation, and valuation, there are many candidate brain regions and networks that are at play here. So my colleague Danny and I, we were saying, okay, well, let's kind of figure out a plausible modeling space, because we knew that we wanted to use SCA. We had a large sample of participants, over 200 who completed this paradigm. And we wanted to kind of sketch out a priori neural indicators as kind of the primary predictors of interest. So again, we had three presumed processes: reactivity, regulation, and valuation, and different brain regions associated with these. But importantly, in terms of how we operationalize the brain activity, we had two ways we could do it. We could look at kind of the average activity in particular regions, or we could look at whole brain patterns. So it's a kind of different way to analyze the data, but it assumes that the brain is kind of carrying out these psychological processes of interest across regions. So that the key level of analysis is not what a particular region is doing, which would be kind of a more modular approach in how we think about brain function, but really it requires a coordination of multiple brain regions, and it's the coordination of those regions as a collective network. That's what's carrying out the psychological process of interest. But we wanted to say, okay, let's look at it both ways. And with specification curve analysis, you can. We had a priori covariates as well in terms of participants' biological sex, the extent to which they were restraining their food intake in general, if they had other goals, health goals, fitness goals that they were pursuing. And then other neural indicators would fall into the covariates bin, because again, for each model specification, we were kind of looking at each predictor separately, and then other other neural indicators would fall into the bin of covariates. We had another way to analyze the data where we could look at kind of their overall brain activity in response to food images, or we could look at their brain activity in response to meals or snacks or dessert. So another kind of granular decision point there. And I'm omitting some details just in the interest of time, but this led to over 200 model specifications, which is a pretty decent number of models to see kind of how the effects are consistent across modeling space. So we were really interested in our a priori neural indicators. And just to kind of lay it out here, we had 8 total, and they corresponded to either reactivity, regulation, or valuation, according to that 3-process model I was just telling you about. And our outcome measure of interest, one of our primary outcome measures of interest, was desire enactment, how frequently they gave into their desires to eat. Now, what this graph is showing is kind of the trace of each of these neural indicators. So there are 8 of these, the trace of each of these across modeling space, right? But if you look at it, like, you can start to discern patterns, but we wanted a way to kind of summarize it and apply the inferential statistics I was telling you about earlier. And what we found was that when it comes to desire enactment, the most reliable indicators were those that were indexing reactivity and valuation. And we did have one region of interest, cognitive control, that was negatively associated with that desire enactment, which was in line with our hypotheses. But you see kind of a range in terms of the median effect size across all the model specifications. So there's a lot contained in this figure here, even though it's just a bar plot. But what's represented by the values and confidence intervals in the bars is the effect of interest for each of these predictors separately across all the different analytic decisions that we toggled in the SCA, right? So it's a different kind of inference, right? You're not just making the inference within a particular model specification, you're making the inference across a broader patch of modeling space. So what can we infer here? We can infer that the extent to which participants spontaneously recruited specific regions and the extent to which they showed whole brain patterns related to the processes of interest, that related to body mass index, which I didn't show you in the interest of time, and daily eating patterns. These results suggest that both uni and multivariate measures of brain activity are valid and predictive of eating behaviors, and the indicators of reactivity and valuation were the most reliable predictors across the different outcome measures. And taken together, these findings can inform future intervention work that perhaps targets how people react to and value food, right, versus strengthening cognitive control. And you may be aware of this, but there's a theory in social psychology that ruled the day for many years, ego depletion, and with a large emphasis on willpower. And as far as these brain data can speak to that kind of controversy, it could very well be that it's more about how we react and value food that could make the largest difference in terms of changing people's eating behaviors. Now, this wasn't an experimental study and we didn't do any training, it wasn't an intervention, but at least some of the results point to that. Okay, last slide. So just to provide some recap and closing thoughts on specification curve analysis, SCA can mitigate issues that arise from researcher degrees of freedom. SCA helps us quantify the consistency or vibration of parameter estimates and effects of interest. And I would like to argue that SCA kind of opens up a totally new and exciting level of analysis and inference. And that is model specifications within a potentially vast modeling space. But, and I don't have a lot of time to go into this, but it's worth noting, this idea of garbage in, garbage out still very much applies here, right? Because if you have poor or invalid measures you're putting into your SCA to begin with, it doesn't matter what you find. Even if you see a really strong effect across all specification, doesn't matter. And Doctors Flake and Freed do a wonderful job kind of highlighting a lot of questionable measurement practices, QMPs, in their recent paper. And as I'm thinking about SCA and how to use it in my own research, it's interesting because I think to some extent, SCA blurs the distinction between planned and exploratory analyses. Or put another way, SCA gives us license to kind of explore and roam the modeling space that we wouldn't otherwise have the opportunity to do. And I'll end with this. I would make this suggestion to your field as well as mine, that we should perhaps normalize or encourage not pre-registration of discrete analyses and models, but pre-register model specifications and specification curve analyses and all the steps, including a priori models that we perhaps think are most important. And that can kind of save us, again, from researcher issues with regards to researcher degrees of freedom. But it's kind of, again, a whole new level of thinking about how we pre-register, design, analyze, and infer results from our studies. And with that, I'd like to acknowledge the wonderful research assistants that helped me here at Bard in the Reach Lab, and my collaborators, Dr. Daniel Cosme and Dr. John Forney, who've been really helpful in helping me think about SCA and the various implications that come out of it. So, and last but not least, thank you all for your time and attention. I'm happy to take questions.
Thank you so much, Richard. That's really fascinating. I've been jotting down questions here as we go through, but I'm not sure if they make sense or not, because I have to admit I'm not a statistical person, so my questions might not make sense. But Vanessa, do you have any preference to open up to in the chat, or anybody have questions here? I think we can just probably.
Yep. I think everybody can unmute and jump in.
Aaron, go for it.
So my first question would be, great presentation first off, but you mentioned at the end there kind of a more widespread use of SCA and maybe even more as a default procedure. What's the computational power required for an SCA?
It's a good question. So as we speak, in the last year, there's been a lot of development of several R packages. One of them is called SPECR, or SPECR, S-P-E-C-R. And I think my collaborator, Dr. Cosme, kind of carried out a lot of the steps for the paper I was telling you about at the end, but there are some limitations in terms of how to work with the functions that are in those packages or change the functions they're dependent on and kind of go under the hood and make some changes. If you have complex models with a lot of covariates and a lot of combinations, it can get unwieldy. I know that my collaborator said to kind of crunch an SCA with on the order of hundreds or maybe 1,000 or 2,000 specifications, it's not too bad. I don't know how that translates in terms of time, but by and large, my sense is that it's relatively easy to implement as long as you don't have too large of a set of specifications. But I'd have to think about that and look into that a little bit more. And that tutorial I showed you, kind of reference from there, Danielle kind of mentions a few of the packages, but I know SPECR was kind of the one that we found most accessible for SCA novices, or people who are not so familiar with it quite yet. That's a good question.
So, but I do want to jump in and just make one little comment. Bootstrapping does take a lot of power, and one of these studies you mentioned, Amy Orban's study.
Yes.
Amy's mentioned to me, or at least to the group with Daniel Lockins, that analysis was they had to rent out the supercomputer at Oxford, and it took about 48 hours for that entire SCA to run. So right. just a forewarning to anybody who's getting into this, like, this is not as easy as running an ANOVA.
No, it's a fair point, and I'd have to check with my collaborator to see. It could have been that we only had, like, we tried to keep it streamlined as possible, and I know that in Amy and Andrew's paper, I mean, they were looking at a lot more because they really wanted to get a fuller handle on those associations. So it'll depend. So like maybe that's a cop-out answer, but it'll somewhat depend. But it's certainly a consideration, a practical consideration for anyone who wants to take this on. So thank you for bringing that up.
Can I go?
Sure.
Richard, again, apologies if this is a stupid question. I'm trying to wrap my head around the kind of exploratory versus planned parts of this. Is it necessary to a priori justify all specifications, or is there some room to just basically chuck everything in and see what happens within specification?
I'm not sure. I mean, I think what's best to pre-register or have laid out at the outset is to make it clear kind of what are your predictors of interest and what are other covariates or other analysis decisions. So at least you're saying this is the set of specifications that it will involve based on that, right? But I think it's okay to be agnostic at that point, especially like the fact that you're doing an SCA, you're not necessarily sure what the effect will look like across modeling space. And I think that's totally fine. Like that's partly the point. So I think pre-registering as many things as possible, but for us, we had a sliver of the modeling space where we had well-defined a priori models. And we said, this is what we would expect in these models. But we also wanted to push the envelope and say, well, those effects may not be reliable or consistent. So we're actually going to kind of map out this whole part of modeling space under these conditions and see if the effect changes and what the nature of the effect is, across those specifications. So I think being, I don't know, I hear a lot of people saying this in kind of the open science community, just be honest and transparent about what you were agnostic about and what you had really good reason and rationale for at the outset. And that, of course, will help a reader or reviewer or whoever kind of upweight and downweight, right? Like whatever results fall out based on everything you kind of laid out at the beginning, and making that as clear as possible.
And that was actually, sorry, that's actually my follow-up question. Apologies, I don't want to take over here, but my follow-up question is it possible to weight within the model based on your kind of the theory or the justification for different specifications, or do you just kind of put them all in as equal and then just interpret them based on theory?
That's a great question. I don't know how weighting would work. I was just describing it kind of just in a general interpretive sense when I was saying that then, but in terms of running the SCA, I'd have to look. I don't know what that would look like in the SCA framework, but that's a really interesting question. Like, yeah.
Do we have anyone else have any questions?
I have another one if there's nobody else. So here we go. So my question would be more of when is this? So I think I'm a little skeptical of this technique, to be honest. I think it could be very interesting for these vibrational effects type of models. Epidemiological models come to mind from as a great situation because there's a lot of throw the kitchen sink type mentality.
Sure.
And so it's good to kind of show these vibrational effects and say like, hey, having one model that has this extreme of a fit isn't unlikely. It makes perfect sense to me. But as a physiologist, I kind of struggle with seeing this more in where I do most of my work's toolbox, when we have really well-defined primary and secondary outcomes, where like I'm even thinking of my study where it's like we have one outcome, that's it. And we have maybe 3 to 4 covariates that will add to the model, and that's it. And we know exactly what type of things. So I guess my question is, if you have a really well-specified model or handful of models ahead of time, is SCA really worth it?
It's a good question. I will say it may have less value in the case, relatively less value in the case you just laid out, but at the same time, as soon as you have like 3 or 4 covariates and you like at least do the combination and say, well, how many possible, like total possible models here, it certainly couldn't hurt. Like in that case, maybe an SCA could be like a supplemental analysis just to kind of bolster everything that you had taken the time to like, you know, define and say, this is what we expect, this is how we're setting up, we have good reason to. So that all that's to say, SCA doesn't have to be the end all be all, and it's going to have relatively higher and lower utility depending on your exact situation. But I certainly think it could not hurt, because again, like something unexpected may pop up from it potentially. I mean, that probability will vary a lot depending on how you set up the model. But again, like which measures you use, and assuming your outcome is like the gold standard, and it may be in the case you just described. But in other cases where it's like, well, we have this gold standard outcome, and we have these 2 or other 3, you know, easier to measure outcomes, like giving people a saliva test, or I'm just making things up, but just like other, right? And then if you're wanting to compare how it does, like looking at a new candidate outcome that works just as like, so then it depends. But overall, in some cases, I would say, yes, it might have relatively lower utility or value, but it could still kind of fill in the gaps a little bit. And in other cases, I would say absolutely, like it's like part of your research question and what you're looking at. So yeah.
I think that's probably going to like put myself and probably focus on more physiology-based research that tends to have an outcome that we're kind of focusing on.
Yeah.
I suppose you're working in a broader space, psychology, nutrition, where you have multiple outcomes and multiple ways to consider things. I can see how it is varying value in those kind of situations, depending on the question.
Any other questions or?
I have one. Andy here. Hello. It was a really good explanation, Richard. Thank you for that. It made me think of a couple of extra things that may have been confusing to some people. I don't know. And I'm not sure I'm going to be able to explain myself too well either. But I'm assuming this isn't a situation where you would run this in order to decide what the best set of decisions was. No, because I could imagine, and I'm not saying that someone should do this. In fact, I'm saying the opposite, that if someone ran it, and then, for instance, on your graph, where if you said within a particular effect variable parameter you chose, if you saw that actually the effect of your decisions was quite steep,
yes,
that someone may decide, oh, well, if I choose this specific set of specifications, my effect size is going to be larger on average, and therefore I should. And I think I almost think it's important to realize that this is something not to make a decision, but to look across possible decisions, right?
That's right. That's exactly right, Andy. I really appreciate you bringing that up. I mean, we can come back full circle, and this is essentially what happens again. And any tool, SCA included, can be mishandled, misused, misappropriated. So yes. So what I would say is, if you unexpectedly came upon, oh my gosh, if we make these, you know, this set of decisions, we're seeing like, but it should be not so much to like mask the rest of the modeling space, but it could actually give you insight and say, well, why is it that the effect either goes to zero here? Because actually a paper that's in its very, very early stages that I'm doing with John Flournoy and Daniel Cosme, we're trying to think of possible scenarios where this exactly would happen, right? Where you have collider effects or other issues of collinearity between predictions. There's a lot of muckiness and messiness, but still, if you really care about truth and trying to find something that you can hang your hat on, you want to know about all of these potential fraught issues, right? That could come out of this. So, but yes, in short, this would be not to be prescriptive and say, okay, then we're going to do it just this way and report it. But you know, if you have another sample, like to do SC, so you could bootstrap to get a little confidence, you know, within a sample. But if you have another sample and you run the SCA again, say, well, you know, will we see that same pattern of this model specification leading to a really inflated or a really minimized effect or something? So yeah, for sure. But that's a great point.
I also guess as an additional point to that, if one of your criteria for your model choices was to do with data quality, that might have such a huge effect on whether that variable was consistent or not, that telling you something different from how consistent it is.
Yeah, and that, I mean, it's a related, but it's a separate issue of just ensuring that your data are as clean as possible in terms of the quality of the data and the validity of those data representing the measures and constructs you care about. And like, you can't get away from that. SCA, of course, will not do that at all.
And that's why you put garbage in, garbage out at the end.
Yeah.
The same thing. Yeah.
Yeah.
Cool. Thanks.
Yeah. I see a question in the chat from Christoph. In my area, there are many options. Do you think it's about on the different processing choices? Yeah. If I'm understanding Christoph's question right, sure. I mean, yeah, because depending on how you process the data, right? SCA can shed some light on the effects of processing the data or cleaning it in one way versus another. It's just a different kind of inference. It's not so much an inference about, you know, the predictor being related to the outcome, but if you see the effect increase with data quality increasing, but again, if you're processing the data in different ways, I'd have to think on that more, because is it that you're processing it in a way to make it more valid, so it's actually hitting on the construct, or you're artificially doing something to it, which is creating a spurious association between that measure and the outcome. So I'd have to think about that more, and you'd have to, yeah, but that's a good question, Christoph, thank you.
I would say, I'm just kind of thinking, like, say, for example, I don't know, VO2 max cleaning. I would say there's probably no one gold standard you must process in this way, in which different people, different perspectives, and again, depending on your question, what you're really looking for, there might be different ways of processing. So I don't think it'd be a case of, I'm just processing, there's no one gold standard.
Right.
There's recommendations. So I think there's where my mind went on that question from.
Yeah, and that actually reminded me of something I wanted to say on this slide when I first presented it earlier, and that is, yes, the total possible workflows, like in theory, is about 69,000. But in terms of best practices and what people do, and just based on other work, that can be reduced down. It's still a lot, like maybe it goes down to 1,000 or something. I'm just making up a number, but if it goes down, but there still is a lot of play. There's a lot of latitude. And you have to kind of then go to, experts in your own field, whether you're doing the work or others, and to try to come around like some consensus to standardize how the data are processed. Now, in some cases, the processing steps will not have an appreciable effect in the end, right? And that's great. I mean, just that, again, like as you process the data, are you, leaving the construct or what it's validly measuring, or are you staying pretty close to it? And that's going to depend on a lot of things, but including how you process the data potentially. But if a data processing pipeline is relatively foolproof, that's great, but it's not always. And certainly in fMRI, it is not foolproof. At some steps, it's relatively foolproof, but definitely not all steps.
Great. So we're just about at the top of the hour. So thank you again very much. It's very interesting and kind of blew my mind. And I'm with Emer. I don't know which questions to ask, but so thanks to everybody that's on the call. And just to know that we're kind of going out of our sequence of every other, that our next one, Journal Club, is April 14th. Am I correct, Emer?
April 14th. Paper, material for us next year is going to lead that one.
And we apologize if anybody got caught up in the time changes that occurred on the Pacific Coast and not in the UK, but we'll work through that as well. All right, so everybody have a wonderful, wonderful day, week, and we'll see you in a couple of weeks.
Bye. Thank you all.
Thank you, Richard.
Bye.